right. Well, so we are live with a micro seminar for September, and I'm really excited to introduce Kristen DeAngelis. She did her undergrad at Harvard and then came to Berkeley. So there's another Berkeley connection here in the micro seminar series where she was a grad student at plant and microbial biology. That's where I met her back in the heady days of the mid-2000s, and her PhD advisors were Steve Lindau and Mary Firestone, and she had some really cool microbial ecology projects there. And then she just told me she did 3 postdocs without moving, because why would anyone want to move out of the Bay Area? There's some other micro seminar people coming up who might be able to share that perspective. And now she's at UMass Amherst, digging into the soil oligotrophy, which she's going to talk to us about today. So, Kristen, take it away.
Okay, thank you. Thank you for that nice introduction. I'm going to head right into my slides. Let's see. How's that? Good? Okay.
Looks good.
Okay, awesome. So thank you for the nice introduction, and thank you for this opportunity to tell you about our work. So my lab at UMass, we are a microbial ecology and molecular ecology lab. We also do environmental microbiology, and I'm interested in climate change and how that affects microbial communities. And so today's talk is going to be more on the microbial ecology side of things, and we're going to be digging into the microbiology soon, and I'll tell you a little bit about that too. So I guess before I move forward, I want to tell you that this work is supported mostly by my startup from UMass, community sequencing project from the Joint Genome Institute, and the continuation of this work will be funded by the TES program at the DOE. We are looking at microbial community response to long-term warming in these soil warming sites at the Harvard Forest. The Harvard Forest LTER, the Harvard Forest Research Station is an LTER site, or long-term ecological research station, and this is an NSF program, and I've been very lucky to have collaborators in Jerry Melillo and Sarita Frey who've been running these experiments for over 2 decades now, and so I'm going to tell you more about the sites and about our microbial ecology, but first I'm going to sort of introduce this problem, which is that the Earth's climate is warming, and this is being exacerbated by biogeochemical and biophysical feedbacks. We know that microbes are key players in all biogeochemical cycles, but the way that microbes sort of scale from having individual responses in the soil to the ecosystem is not well understood, although we know that this does happen. So what I'm showing you in the slide are 3 examples of key ecosystem processes that are driven by microbes and ways that microbes might have positive or negative feedbacks to the climate system. And so despite their important role, we're only beginning to sort of understand how to incorporate microbial processes into models. Biogeochemical and biophysical feedbacks. We know that microbes are key players. Sorry, somebody, thank you. Okay, I was getting some feedback. So at this broad scale, at this ecosystem scale, we know that climate warming speeds metabolism respiration of microbial communities and results in accelerated loss of carbon from soil. So I'm showing you a figure from this nice meta-analysis of many studies of climate change studies that had occurred around the world, and that's the map on the right. And what you can see in this table is that in parentheses are the number of studies that went into looking at what the net effect of warming was. And you can see that there was a net positive effect or net increase in above ground biomass in response to warming, a net increase in total net primary productivity, a net increase in below ground net primary productivity, and also a net increase in ecosystem respiration, above ground respiration, and soil respiration. So at this sort of ecosystem scale and around the world, all soils sort of behave consistently in having accelerated respiration. And in fact, many of you microbiologists will also know that if you grow an organism at a temperature slightly higher than the one that it had been isolated from, it will grow faster. Usually the temperature optima are higher than where the organisms are used to. So over the short term, warming speeds metabolism. So we wanted to look more closely at the long-term warming studies, and specifically ones that had been running for 3 years or longer, and only at the studies that incorporated some component of microbial community composition, microbial biomass, or microbial activity. And of the dozens and dozens of warming studies that have gone on for more than a few years, very few of them, relatively few, are looking at some component of microbial communities beyond just measuring respiration. So that's the first point I want to make here. But the second point is that the long-term warming effects are ecosystem dependent. And so that really necessitates sort of looking at warming effect on microbial communities in different ecosystems in order to properly go from this ecosystem scale to a global carbon budgets and incorporating microbes in a meaningful way. Sort of the most in-depth microbial analysis happened in this tallgrass prairie studies in Oklahoma, and they were able to show that ultimately it seemed as though warming in the tallgrass prairie systems would result in a net negative feedback to climate. Our preliminary data suggests that in the temperate forest soils, microbes are going to be net positive feedbacks to climate, and I'm going to tell you more about that now. But before I do, I want to sort of remind you about microbes in soil. And this is where we're thinking about microbes. There are a lot of niche spaces that support a lot of diversity in soil, and that is because of different size aggregates, microbes living within and between aggregates, soil pore water that is to varying degrees continuous or discontinuous with the soil water as a whole, and so you have these small regions where you can have anaerobic metabolism, microbes that are exposed to very different redox potentials, very different solvent concentrations, and very different nutrient concentrations. And so theoretically, there is a range of nutrient availability in any soil. But when we think about trophic strategy, and trophic strategy as a definition is just a way that an organism decides how to allocate limited resources to grow optimally. And so that might be fast growth, or that might be slow growth, or that might be ability to use very high concentrations of nutrients and withstand high osmotic potential, or use very low concentrations and make the transporters that are required for that. So microbes that are adapted to this low concentration or poor nutrient availability are oligotrophs. High concentration are copiotrophs, and it's not really a dichotomy, although when you start looking at the genes responsible, there is a genetic basis for trophic strategy, and this genetic basis for oligotrophy in soil is sort of what we're interested in. We're interested in this with respect to this long-term warming because I sort of told you that our forest soils are net sources of respiration under warming. And so this is a picture of our study site. It's the Harvard Forest long-term warming site at the Prospect Hill. And we're not studying warming in winter, but I like this winter picture because it gives you sort of a view of the scale of the warming experiments, and you can also see the warming effect in this picture, which is nice. The Harvard Forest LTR is located in central Massachusetts, just sort of northeast of the Quabbin Reservoir, which is in New England. This experiment has been going on for 20 years. And what I'm showing you here are 3-year running averages of the carbon released in the heated compared to the disturbance control sites. Now, I forgot to mention this in the previous slide, but the way that the plots are heated are by these below ground resistance cables. And at the time that the experiment was set up in 1991, there were established the heated sites, disturbance control sites that had cables installed but never turned on, and then control sites that were undisturbed. And there's been no long-term effect of the disturbance, but we are using disturbance controls as our controls for the rest of the data that I'll show you. And so what you can see here is that over the course of the first decade, the Harvard Forest field study behaved much like all the other field studies, for example, that were summarized in this Wu et al. Nice meta-analysis. There was a lot more carbon released in the form of CO2 in the heated compared to the control sites. After about a decade, this treatment effect went away, and there was no significant difference in the heated compared to the control. And now I'm sketching out the data because this is not published, but this is real data. And there are some reasons why we think this treatment effect went away in the second phase of the experiment. First of all, I should tell you that 70 to 80% of the respiration in these soils are microbial, the remainder being root respiration. It was observed that there was thermal adaptation of the microbial community respiration in these warming soils. In other words, the microbes became more carbon use efficient at higher temperatures after this decade of warming. There were also observed decreases in labile organic carbon after this first decade. So this is sort of the easily available carbon or substrate for the microbes that after this amount of warming was being used up more quickly than is being replaced. And after 2 decades, it has recently been observed that the soil organic matter is becoming significantly depleted. So there's a lot of loss of carbon from the soil. Sri DeFray's lab showed some microbial community analysis using fatty acid methyl ester analysis that there's decreased fungal biomass with warming, along with a shift in the mineral soil microbial communities towards increased abundance of gram-positive bacteria and increased abundance of actinomycetes. And at the time that I was coming in, this was sort of the bulk of the microbial community information that we knew about these sites. So I told you about this phase 2 in soil respiration. What's happened in the last 7 or so years is we've entered into this phase 3 in this long-term warming sites, where there is now more respiration in the heated soils compared to the controls. And now we know that the labile carbon, or the substrate that the microbes have been using, is depleted. We know that there was thermal adaptation 10 years ago, and we know that there is somewhat of a community shift. And this is data looking at microbial growth efficiency after 18 years of warming on the right, and a companion study that was started and at the time of this publication 2 years prior. And so that's on the left. And what this is showing is that when you add glucose, which are the graphs on the top, there is not an appreciable treatment effect if you incubate the soils at 5, 15, or 25 degrees. However, if you look at the bottom graphs, you'll see that if you add phenol as a substrate, this being like a model recalcitrant substrate, there is decreased efficiency at higher temperature, but that effect goes away in the heated soils. Now I'm pointing you to this bottom right hand, and this big difference at elevated temperature where the microbes seem to be more carbon use efficient at higher temperatures. but only for this recalcitrant substrate. So all of this data taken together has led us to this hypothesis, which sometimes we call the recalcitrant decomposer hypothesis. And it's this idea that over the course of long-term warming, microbial communities have shifted towards ones that are better able to decompose or access recalcitrant, lower quality, or less available carbon. And this is consistent with this idea of oligotrophy. And so, as a microbiologist, I'm interested in trying to understand, how the substrate availability has changed, but also how the substrate utilization has changed. And we're going to test this. I'm going to show you data today from the soil warming Chronosequence. Okay, so what's that? I alluded to the fact that there were multiple warming experiments, and there are 3 that we're calling this warming Chronosequence. These are warming experiments that were started in 1991. This is the respiration data that I was just telling you about. A second study started in 2003, and a third study started in 2006. At the time of sampling for the rest of the data that I'm going to show you, these experiments were running for 5, 8, and 20 years. The plot setup is a little bit different for each one, but they're all heated in the same way, 5 degrees above ambient by these buried resistance cables. And this is just to remind me to tell you about that this middle study is special because this is the only study that is set up in such a way that we can look at the plant effects of warming and root inputs into the soil. So a lot of the labile carbon coming in is going to be through root exudates, but in the smaller plot sizes, the plots are set up such that they avoid trees, and so only partial root masses would have been warmed, and we just don't know how the tree would respond. But we can use this Berrywood study, and I'm going to come back to this at the end. We can use this study to make some inferences about what's going on with this plant microbial interactions. And so I told you that there are 3 experiments. We're also looking at 2 soil layers. We're looking at the organic layer of soil and the mineral layer of soil. These are distinct layers. And you can just see that they look really different, and all of our data bear this out. Now, the resistance cables, the heating is happening in the mineral layer of soil, but we feel confident that we're getting a uniform distribution of heating, and there's other data from other warming studies that support this. And so this is our approach. Our experiment includes 48 samples with the 3 sites that I just described to you. 2 treatments, the heated and disturbance control, 2 soil types, this organic horizon and the mineral soil to 10 centimeters depth. We have 4 biological replicates so the data that I'm going to describe to you now are results from a bacterial small subunit ribosomal RNA gene study that we did some sequencing using the Illumina MiSeq. We're thinking about the fungi, but this is the sort of domain of Sarita Fry's lab, and so I'm not going to talk about the fungal community today. We're just focusing on the bacteria. I'm going to show you some quantitative PCR of differences in genome equivalence at the domain and phylum levels for the soils, and I'm going to show you some metagenomic data that we have with a specific focus on looking at oligotrophic markers and plant and bacterial genes. We also have metatranscriptomic data, and that is coming soon, but I'm not going to talk about that today. So we are doing ribosomal RNA sequencing on the V4 region using the human microbiome protocol for MiSeq, which was really successful. And we, just to remind everyone, we like the ribosomal RNA gene as a phylogenetic marker. It's wonderful. It's highly conserved with variable regions, so and it's a structural RNA, so it's very information dense. It's short, so it's amenable to this sequencing. It's cultivation independent. The databases on 16S are very, very rich, and 16S copy number is related to trophic strategy, and I'm going to talk about that a little bit more later. But right now I'm going to tell you about our data. So we did this MiSeq run. We had about 11 million reads. After quality control, assembly, removing spike in reads, we ended up with about 3 million reads. We elected to exclude singletons and doubletons. We removed chimeras. And these 3 million reads were binned into 45,000 operational taxonomic units. So taxa classified at this 99% identity. After rarefaction, this went down to about 43,000. And when appropriate, I'm going to show you the rarefied data. I was also interested in this dominant subset community, and as an arbitrary cutoff, and I know that there are some better ways to do this, but we elected to look at the taxa that were abundant at this 0.1% relative abundance or greater, and that left us with 155 OTUs, but these accounted for half of our observations, which I found really amazing in soil, because there is so much diversity, but they are actually really uneven. So what I'm showing you now is the 16S data, and this is unifrac distance measure, which is a phylogenetic measure of how different the soils are. And I actually did one principal coordinates analysis, but I split it out into 3 panels so that you can see the treatment and soil effects in the different sites. The biggest effect on the microbial communities is soil type, and we probably didn't need to do a ton of sequencing to know that soil type is the biggest driver of differences in microbial community. If you remember that picture, it would be obvious. The other interesting thing, though, is that I spent some time in the introduction telling you about all these differences that we had observed in acclimation of respiration and carbon use efficiency for different types of carbon utilization that were happening much earlier than 20 years, but we don't see a treatment effect of heating until the 20-year site. So it's almost as though the microbial community phylogenetic profile has changed last. And that might not be so surprising when you consider that something like 10% of the community in soils is supposed to be active at any one time, because there are so many places to hide. Soils can support a very high amount of high fraction of dormancy. And so I'm thinking that just changes in relative abundance or changes in the active fraction would have partly been responsible for some of these functional changes that we had observed earlier. So we also did metagenomic data analysis. This is through the JGI. We had 1 lane of high-seq per sample, so this is an average of 7.8 million sequences per sample. Most of the sequences were bacteria, with the other domains represented, although I think that this is partly a reflection of just the data that's in the database for doing the annotation. But what I can show you is that if we look at the sort of functional communities based on the metagenomic data, and this is based on enzyme classifications using Bray-Curtis dissimilarity, we see that we can see changes in the organic soils much earlier. So the symbols are a little bit different, but here is the heated, and here's the control for the organic soils in the berry woods, and this difference is significant as well as this. And so that suggests that there's a difference in functional potential that happens earlier than this phylogenetic difference. So who's there? We're going to go back and talk a little bit more about this MiSeq data. I'm showing you a rank abundance curve just for the top 155, and because it's ranked, there's this very long tail of diversity. And there are a few taxa that really overwhelmed our measure of abundance. And this inset is showing you the 4 taxa that are the most abundant. So we had 2 actinobacteria, one, this is the most abundant, that decreased sharply with warming, one that did not change, an alpha proteobacteria that increased dramatically with warming, and an acidobacteria that did not change. And so we wanted to also look at, so this is sort of a window into who are the most dominant taxa. We can also look at the phylogenetic distribution at the phylum level, both for the 16S, which is here on the left, and the metagenomics on the right. And they both point to a dominance in the soil communities of alpha proteobacteria, actinobacteria, and acidobacteria, where this red is all proteobacteria. This is just proteobacteria on the right, but this red, again, is the alpha proteobacteria. And what I want to mention now for the future of this talk is that you should recognize that alpha proteobacteria, actinobacteria, and acidobacteria all tend to have a low copy number. They tend to be more considered to be oligotrophs, and so this is perhaps the first clue that this is that this is something that's going on. So we did quantitative PCR on the fungi and bacteria for all sites, and this is on the left. And we found a near significant decrease with warming in the fungi compared to the control. And this is consistent with Sarita Fry's FAME analysis that showed that heating had a significant effect on decreasing fungal abundance in the heated soils. There's no difference in bacterial genome equivalence by treatment. However, we look at our 18 phyla, these actinos, alpha proteos, and acidobacteria that are among the most dominant. We see that there is no phylum level effect in the abundance of actinobacteria. There's significantly more alpha proteobacteria in the heated compared to the control, and there's this trend towards more acidobacteria in the heated compared to the control. So because the most dominant actinobacteria had this significant decrease in the heated, I felt like I wanted to sort of look at the community data more with an eye towards what the individuals are doing. So how are individual taxa, and who are the individual taxa that are driving this treatment effect in this Prospect Hill organic soils? So we did an indicator species analysis, and an indicator species is any species that defines a trait or characteristic of the environment. In this case, we're looking at species that define warmed or controlled sites in the Prospect Hill organic soils. We actually did 7 tests, and our data fit the assumptions for all of these tests, and we called an indicator species significant if it was significant by 2 or more of these tests. And we decided to do it this way as a way of sort of narrowing down only the most significant taxa. And so when we do this, out of all of our 45,000 species, we end up with about 17 taxa that are indicators of the warmed soils or controlled soils. And so the acidobacteria that were indicator species were all indicators of warmed soils. And you'll remember that we saw a sort of a near significant increase in the total amount of acidobacteria. And this is interesting. Many acidobacteria are known to be able to grow on complex polymers, including cellulose, hemicellulose, and chitin, and perhaps this decrease in labile carbon availability has given these organisms an opportunity to thrive in this low carbon available soils, the warm soils. Interestingly, the actinobacteria had 2 indicators that were indicators of warming and 2 that were indicators of control. Now, in the qPCR, we saw no net change at this phylum level for actinobacteria, but there's something else going on where we have 2 strong indicators in either direction, and one of these is this most most abundant taxa. For the proteobacteria, we found all of the alpha bacteria that were in the order Rhizobiales were indicators of warmed soils. Rhizobiales, of course, have many members that are plant-associated, but they also include many sulfur and purple sulfur and non-sulfur bacteria. So organisms that are able to grow under low energy, low nutrient conditions. The two alpha proteobacteria that were indicators of control sites were both in the Rhodospirales. And so what I can show you too is, and now I know that this is tiny and we're going to zoom in on this, but this is a phylogenetic tree of all of the dominant taxa. And I wanted to show this to you because I want you to sort of see the phylogenetic distribution of these indicator species. And so here are the acidobacteria, and the stars are the ones that were significant by our 2 of 7 tests. And what you can see here is if you look at the actinobacteria here in green, you can see that there's no phylogenetic clustering of the taxa that are indicators of control, which is blue, or indicators of heated in red. Whereas for the alpha proteobacteria, here in this red bar, you can see that there is very good clustering. Here are the Rhodospirales, here are the Rhizobiales. And so, you know, I realized that the 16S is, you know, you cannot infer function from phylogeny, even though we like to sort of play this game. And we are in the process of isolating members from the A team, from both the heated and control sites, with the intention of backing up some of this hypotheses that are generated from this data with physiology studies and comparative genomics. That's the plan anyways. So we've got something like 75 isolates in culture right now, and so this small culture collection is growing in our lab. So I mentioned earlier that ribosomal RNA copy number is related to trophic strategy. There is a phylogenetic signal to ribosomal RNA copy number. Ribosomal RNA copy number is also related to genome size, and it's also related to maximum growth rate. And so this sort of feature of ribosomal RNA operon copy number is also a huge caveat to these small subunit studies, these phylogenetic studies, because your data is going to be polluted by copy number, where if you have taxa that have high copy number, they're going to be artificially inflated in your data. And so there are a few tools now that are available to correct your data for copy number. And we use this one that was published in PLOS Computational Biology. And the way that this works is through phylogenetic inference. So along with this paper, there was published a phylogenetic tree of something like, I think it was almost 700 organisms with the copy number mapped onto the tree. And so this is sort of what I'm showing you here, where species A has 10 copies, species B has 6, and this is actually the same tree that I showed you in the previous slide. And what you do is you insert your own sequences into this tree and use this algorithm to infer what your copy number would be. So then you have your own tree that is a map of copy number to phylogeny, and you can use this to correct your own data. And the authors of this paper, Kemble and colleagues, found in their samples, which came from gut samples, from insects, and something else that were not soils, they found a very big difference in the corrected versus uncorrected communities. We saw no difference, and that is partly because of this. The vast majority of our 16S copies were 2 or less. So there just aren't that many organisms that have high copy number, and the ones that have high copy number are very low abundance. So the copy number correction made no difference to our conclusions for our soil communities. However, we can still use this copy number estimation to look at how the treatment is affecting copy number in our soils. And what we found was that in the Prospect Hill organic soils, there's a significant decrease in copy number in the heated soils compared to the control, whereas in the mineral soil there was no significant change. And so, this is another, at the time that I saw this is another sort of piece of evidence that pointed to this possibility that long-term warming is creating oligotrophic conditions for microbial communities in the soils. So now I'm going to turn to the metagenomic data and start talking about looking at genetic markers of trophic strategy. And I was really excited about this paper, which looked at genetic markers of trophic strategy in marine bacteria, starting off with the model copiotroph and model oligotroph. Trophic strategy is associated with growth strategy, so either feast or famine growth or equilibrium, cell size, maximum growth rate, and some other features, some other functions. Also in this paper is a very nice and long list of clusters of orthologous genes that they found in this comparative genomic analysis that should be indicators of copiotrophs and oligotrophs, and we found very few of them either in our data at all, and the ones that were present were not different. And now, because of the previous data that I showed you, especially with the copy number, we're expecting to find some markers of trophic strategy. And so I put this here just to sort of mention that the markers of trophic strategy in marine organisms seem to me like they may be different than the markers of trophic strategy in soil organisms. And that is a story waiting to be told. And we are definitely interested in that. And if you are, you should let me know. We looked at one effect on oligotrophy in terms of altered central carbon metabolism. And so the Embden-Meyerhoff-Parnas pathway is glycolysis, and 2 ATP are produced per glucose. And this is really important to anaerobic organisms and organisms in low energy environments. However, throughout the bacteria and in some archaea is the Enter-Deuteroff pathway. So this is an alternative glycolysis pathway that seemingly strangely only produces 1 ATP per glucose. But what Flemholz and colleagues did was they looked at the protein requirement for the Enter-Deuteroff pathway and found that this pathway requires 7 times lower protein input. And so they suggest that in environments where energy is more abundant, the Enter-Deuteroff pathway is a better sort of resource allocation. So it's a trade-off. So we looked at 3 genes that were specific to either Embden-Meyerhoff pathway or Enter-Deuteroff pathway to see if there was a difference in abundance in our heated compared to control. And the 3 were all significant. There were a higher abundance of Embden-Meyerhoff genes in our heated compared to control, and there's lower abundance of Enter-Deuteroff pathway in our heated compared to control. And so this suggests that heating is shifting the prevalence of genes associated with central carbon metabolism towards this mode of glycolysis that is more important for low energy environments. This also could indicate a difference in nutrient availability or nitrogen availability that might go along with the Enter-Deuteroff pathway. So this is sort of the last bit of data where I'm going to show you some data just from the Barry Woods site for this metagenomic data. And we are focusing on this site because this, if you remember, this is the plot that is the large 25 by 25 meter megaplot with subplots where whole tree root masses are heated along with the soils. And I wanted to start looking at genes or cogs that were that we annotated to be associated with plants or with bacteria as a way of kind of looking at how plant microbial interactions are changing over the course of long-term warming. And so this picture in the middle, what I'm showing you now, is kind of a summary about what we know from biogeochemical studies of what's going on in the soil. So there is some increase in root respiration and major increase in microbial soil respiration in the heated compared to the control. The heated soils have a significant decrease in fine root biomass that should be associated with a decrease in root exudates and is supposed to be at least partly responsible for this observed decrease in labile carbon availability. We also had observed an increase in nitrogen availability and nitrogen mineralization. And now these two are, of course, related. And it seems likely that because there's more available inorganic nitrogen, plant root allocation can be decreased, while at the same time plant root biomass has been increased over the course of this long-term warming. So big changes underground. So what I'm going to tell you now are about the plant cogs and bacterial cogs that were different, and I'm showing you this ratio of warmed versus control, and these were all significant to a p-value of less than 0.01. There were twice as many plant cogs that were overabundant in the heated compared to the control, and twice as many bacterial cogs that were decreased in the heated compared to the control. And this is also just a time for me to remind you that these are not transcripts. These are genome copies. So changes in populations or changes in sort of gene allocation towards the potential for these functions over 10 years of warming. So first I'll show you the plant genes. And the big picture is that the plants are doing really well. So glucose 6-phosphate isomerase, which is the second step in glycolysis, pyruvate 2-oxyglutarate dehydrogenase, this is a TCA cycle, lots more ribosomal proteins in the heated compared to the control. So more growth, better growth. The exception was this significant decrease in glucose 6-phosphate 1-dehydrogenase. This is the pentose phosphate pathway. This is alternative to glycolysis and would suggest a decrease in anabolic versus catabolic glucose utilization. Looking at nitrogen and nutrient accumulation genes, significant increase in transporters for nitrates, amino acids, metals, phosphate. So from the plant picture, this gene allocation is the plants are doing really well and importing more nutrients from the soil. The bacterial story is a very different picture. So we see with the heating an increase in a bunch of genes that are associated with alternative energy pathways or energy pathways that would be suggestive of poor carbon availability. So archaeal adenylate kinase, which converts ATP to a regulatory signal for changes in gene expression, the pyruvate synthase, this is also indicative of a need for energy conservation, trehalose maltose hydrolase, this is glycoside hydrolase family 65, so enzymes that would be associated with degrading more recalcitrant carbon and polymeric carbon, cellobiohydrolase, endoglucanase. Allantoicase is associated with nitrogen mineralization. Cytolysin is a secondary metabolite that bacteria make when they're competing with each other. So these all suggest that the heating is creating a need for more different enzymes that are able to degrade more polymeric carbon. The exception is N-acetylglucosaminidase, also known as chitinase, so less chitinase genes in the heated soils compared to the control, perhaps a side effect of decreased fungal abundance, and a significant decrease in a bunch of different phage proteins. This could, I'm not quite sure what to make of this. This could be a result of this changing relative abundance of different bacterial populations, and it could be as simple as the host of this phage is missing. in the heated soils because their niche is missing for other reasons. So I will stop now and tell you our conclusions. So we used MiSeq and community analysis and metagenomic analysis to show that bacterial communities change with warming, but long after these functional changes are observed. We think that long-term warming is encouraging oligotrophic strategies, including a decreased ribosomal RNA copy number, pathways that are shifting towards ones of lower energy environments, and other gene signatures of oligotrophy and stress with long-term warming among the soil microbes. And sort of looking forward, you know, we recognize that oligotrophy is a functional strategy, and one of the things that we're really interested in looking at is testing this functionally and looking at carbon use in soils from the heated control. We're doing some of those experiments now. Once the organic layer is lost, because there's this net decrease in organic carbon, how will the mineral soil act? So most of the changes that we observe were in the organic layer, which is rapidly being depleted. So there's more to the story, I think, and at some point we're going to lose the organic layer completely, and that will be interesting. What is the basis for this community adaptation or acclimation or change? And specifically, how have the individuals changed? And I told you that we are doing a lot of cultivation right now of bacterial isolates, and Sarita Fry's lab is actually also doing cultivation of fungal isolates to really get at this question. And the last question, I mean, it's not something that we can really answer, but it's something that I think about is what is the future for the soil microbial feedbacks to the carbon cycle and the climate system? You know, we saw this sort of three phases where there was a lot of respiration and then no treatment effect and a lot of respiration again. And so, you know, maybe that pool of carbon will become depleted and there'll be another period of a no treatment effect while the microbes regroup and decide what else they're going to eat. And this could sort of go on potentially until a lot of the carbon is lost. And so I guess this is just to sort of say, stay tuned. And with that, I will conclude and thank my collaborators. I feel so thankful to have been part of a collaboration with Jerry Melillo and Sarita Fry at the MBL and UNH. And working with Will Werner, who is Jerry's technician, and Linda Van Diepen, who was a postdoc, and I think now she's a staff scientist in Sarita Fry's lab. None of this would have been possible without them, and I'm so grateful for them for maintaining these wonderful sites over the years. I also am working with Jeff Blanchard at UMass, and a lot of the work that I showed you was the result of analysis by Grace Pold, who's my graduate student, Begum, who was a rotation student in my lab, and William, who is a graduate student in Jeff's lab. And we also had some help from some wonderful summer students and summer REU students. And with that, I am going to finish my talk, and I will be happy to entertain questions. Thank you.
Thank you, Kristen. That was fantastic. I'm going to open up the microphones for those of you who are in here, so you guys can feel free to ask questions if you like. And then we have at least one question on Twitter already from Laurel, who's actually in the HOA. Do you want to just ask in person, Laurel? Oh, I can't hear you. There you go.
Are you there? Oh, well, will there eventually be no organic horizon? Is that what you're talking about, Cameron?
Sorry, can you say it again?
Oh, so I tweeted about how you were talking about how the organic horizon is being lost more quickly than it's replaced, and whether or not we will eventually have just no organic horizon. And so I guess, how quickly is it being lost? Is it the kind of thing where it will be there in the fall and by the next spring there's bare mineral?
Well, so the Melillo lab has done these measurements, and what's happening in the heated soils is that they're starting to become these patches of just bare mineral soil. And they did measurements from all around the plots and found that the thickness of the organic horizon layer is depleting. So it's a loss of total carbon, but it's also a loss in like the thickness of this layer. So if anybody has ever been to a tropical forest, you'll know that the microbial activity is so fast that litter falls from the trees and is immediately respired. There's no organic horizon because there's so much microbial activity. And so this is where it looks like these soils are going, is to this point of the organic matter being depleted much faster than is being replaced by leaf litter deposition. And now, there's going to be more to this story too, because climate change is going to change plant community composition, which could change the chemistry of the leaf litter that's being deposited. So I guess it's complicated. Does that answer your question?
Yeah, definitely. I'm curious if you're seeing phosphorus limitation, or if other nutrient limitation is starting to crop up.
I don't know about phosphorus limitation, but from the plant perspective, there's more nitrogen available over the course of the warming. So I don't know about phosphorus. I'm going to think about that, though. Yeah, sorry, I can't. I don't know if I can answer the question any better.
No, it's definitely one of the things to think about in environments like that, especially in the Northeast, where there's enough nitrogen that it could be saturated. And as we start, we have a lot of nitrogen and less carbon, and the phosphorus story there seems like it could be pretty interesting.
Yeah, definitely. I mean, it does seem like phosphorus limitation is important in older soils and soils that are more weathered. So if you think that warming is accelerating soil weathering, then that should become more of an issue.
Yeah.
Look at that.
Okay, there's a couple questions on Twitter. The first comes from Ashish Malik. I might have to ask you to clarify this. Maybe Kristen will pick this up. The question is, the A-team allocation to oligo or copiotrophy is controversial. Some consider actinos as copiotrophes. Any comments?
Oh, yeah, absolutely. I agree completely. In our soils, so I guess all I'll say is, in our soils, the A-team members had low copy number of ribosomal RNA. How that translates to copiotrophy or ability to use more recalcitrant carbon or more polymeric carbon, or to grow under concentrations of carbon that are lower and nutrients that are lower, is definitely an open question, and this is why I mentioned that we're doing physiology. So I agree that it is controversial.
Okay, and from Leanne Benning, why so few eukaryotic signatures, such as mycorrhizal symbionts, etc.?
Yeah, this is a great question. So we looked for mycorrhizal signatures, gene signatures in the metagenomic data, and did not find any based on this pipeline annotation that came both from the JGI, and we annotated our metagenomic data through MGRAST. There are only a couple, I'm going to get into trouble here, one or a few full genomes of mycorrhizal fungi. And so what we need to do is use those genomes to pull out sequences that perhaps were not annotated because they're just not part of this database. And this has been a theme, actually, in the metagenomic data, which is that the annotation for the bacteria is very good, and there's a lot of eukaryotic diversity that we're just not able to do a good job of analyzing right now because those genes just aren't represented in the database. So microbial, eukaryotic microbiologists sequence some genomes for us.
Cool. From Javier Izquierdo, do you know if soil structure, micro and macro aggregation, is changing in the organic layer of these sites?
Sorry, can you answer the question again?
Yes, it's do you know if soil structure, such as micro versus macro aggregation, is changing in the organic layer of these sites?
This is a great question, and actually one that I really, really want to answer. And I'm trying to get some funding to do this because there are some really wonderful tools right now that will allow us to sort of map the carbon and map the microbes onto the soil structure and look at changes in soil structure. And so I love this question. I cannot answer it, but like maybe hopefully in a year or two I will be able to, because it definitely seems like that is going to be in effect, and I don't know, but like great question. And hi Javier, nice to get a question from you.
Cool. I have a question while we're waiting for any others to pop up that concerns just general sort of genomic signatures of oligotrophs. Have you guys done, or are you planning to do, any sort of average genome size assessments to see if things are smaller than normal for things like in the rhizobiales? I mean, I guess some of these organisms that might be popping up might be unique versions of ones that are larger genome size, have larger genetic complements, etc. And but nevertheless are still nested phylogenetically within those groups. And I'm just wondering if you're seeing unusual genomic signatures for the phylogenetic placement of these organisms that might be more supportive of oligotrophic habitats.
That's a great question, and one that we have not attempted to do yet, but because the metagenomic data is really new. Average genome size. I think that's definitely something that we should look into, and I have not done that type of analysis before, but can you do it the same way as with the copy number, like by phylogenetic inference? Is that the idea?
Essentially, it's based on measuring the number of times that you see certain things like single copy marker genes, and then depending on the method, running some statistical normalizations to make sure that you're doing everything correctly. There's 3 or 4 ways to do it out there in the literature, but it essentially is just looking at whether or not you're seeing things going towards these more streamlined genomes that we typically see in oligotrophic marine environments, I guess is what I'm getting at. Like, is that happening in soil? And what do those organisms look like? I don't. Maybe I'm biased, but I don't think of 3 to 5 kb genomes as being signatures of oligotrophs. But if they're typically 6 or 7 kb genomes in that group, they definitely could be streamlined relative to others, right? So there's no reason why you can't have those effects happening at any genome size. And I'm just wondering if this might be a killer place to look for that, especially since you have whole lane of HiSeq per sample.
Yeah. Yes. Well, let's see. I guess there's two things that I want to say. In addition to saying that, like, yeah, we definitely want to do that. Thinking about comparing 20 years of soil warming to millennia of marine microbial adaptation to low nutrient environment, it's really hard to even think that they would, become, that 20 years would be enough to see an effect like that. However, you're right, like we do see some other indicators, so we'll definitely look at that. I'm writing now. This is like a to-do list for us, which is going to be great. The other thing that I'll mention is we're trying to look at this question in a bunch of different ways. So I mentioned that we're doing the isolations, and at some point we want to be able to do genome sequencing of these representative isolates and use the metagenomics and use the MiSeq to map our isolates back into our environments. There's also another project happening in the lab right now where we're going to try and use a couple of these tools available to reconstruct some genomes from our soils. I feel like even a couple years ago, this would have sounded like a crazy endeavor, but I went to ESA this year, and people are doing this now, are reconstructing whole genomes from metagenomic data, and we are optimistic that we have enough data and that the communities are uneven enough that we'll be able to do this for some of our more dominant taxa. So this is the thesis project right now of Dan Burkhart, who is an honors microbiology, like, awesome student in the lab. So hopefully by the end of the year we'll have at least some really long contigs, if not hopefully some full genomes, and maybe I can get back to you.
Cool. Very cool. Any more questions for Kristen? Cool. Well, as always, this will be archived on the microseminar website at microseminar.wordpress.com. And if you have any questions after reviewing it, or if you're just finishing watching it now sometime in the future, hello to future people, feel free to get in touch with Kristen at any point.
Yeah, I agree. This was a lot of fun, and hopefully I'll make some new friends.
Yeah, and how can people get a hold of you, Kristen?
So my email is kristend@umass.edu, and I'm on Twitter at KristenObacter. Both are good ways.
Sweet. Okay, well, thank you very much. This was outstanding. Excited to have this in our archives, and look forward to seeing more from you in the future.
Thank you. Thank you so much.